Okay, so today I'm going to present an update on our current workflow where we're trying to identify potentially new biomarkers that can predict immunotherapy response for triple negative breast cancer. So our lab at the ONJ-CRI is really focused on TNBC, which represents 10 to 15% of all breast cancer patients, particularly metastatic TNBC, because metastasis is what's responsible for the majority of deaths that we still see with breast cancer. So triple negative, which is lacking those three hormone receptors, often metastasizes to the lung, bone, liver, and brain. And because these patients don't have these hormone receptors, they've been limited for a long time to basically just chemotherapy as a first-line treatment. So it was really exciting in 2022 when the TGA approved immunotherapy for use in Australia. So this is pembrolizumab, anti-PD-1, which was approved for use in combination with chemotherapy for both high-risk early-stage TNBC and also metastatic TNBC that expresses the ligand PD-L1. And it was even more exciting when in 2023, pembrolizumab was put onto the pharmaceutical benefits scheme. So that has really improved access for TNBC patients. So what do we know about predicting immunotherapy response in TNBC? So we know that a high level of tumor infiltrating lymphocytes, so T cells, is associated with both improved survival and also neoadjuvant chemotherapy response in TNBC. And also that PD-L1 expression is associated with immunotherapy response, but only in the metastatic setting, so not in patients who receive their immunotherapy prior to surgery. So we currently still don't have any good biomarkers that can predict immunotherapy response for early TNBC. And as such, it's only about 50% of patients who will have a good response, which is still a lot better than what it used to be with other checkpoint inhibitors in trials, but it means that we're still taking a gamble. We don't know which patients will benefit. So we really need to work on what the barriers are that are preventing the activation of T cells in tumors and a subsequent immunotherapy response. And the way that I came into this project is that I've long been interested in the immunosuppressive microenvironment, particularly myeloid cells. And we would have probably all seen this diagram before about hot and cold tumors. So hot on the left, high T cell infiltrate, likely to respond to immunotherapy. But on the right, you have what is known as a cold tumor, which has a balance more towards a higher myeloid cell infiltrate. So myeloid cells can belong to this class of cells called MDSCs, and they are called this because they're really potent suppressors of T cell activation and proliferation. And we only find them in disease settings such as cancer and inflammation, but it's been the sort of confusion between these cells and normal myeloid cells that's led to a lack of targeted treatments against them. And this is just to give you an example of some results from the lab characterizing myeloid cells in preclinical models of TNBC. So we see really substantial heterogeneity just in myeloid-enriched samples from these primary tumors. And we see just even 4 or 5 clusters of granulocytic neutrophil-like cells alone. And so this led us to ask, has myeloid heterogeneity been properly captured in TNBC patient tumors? And the answer was no. Neutrophils have really long been underrepresented in all single cell data just because they're not transcriptionally very active and they can be very sensitive to processing. But we didn't want to just limit ourselves to this myeloid compartment. So we thought about using spatial transcriptomics to better capture the complete whole sort of tumor microenvironment. And in doing this, could we then identify potential new biomarkers for immunotherapy response? And we chose spatial because we know from the clinic and the literature that it's the location of immune cells such as TILs within the tumor that can really impact their prognostic significance. So this project started a really long time ago before immunotherapy was approved for TNBC in Australia. So in terms of accessing patients, we were really limited to clinical trials. So breast cancer trials granted us access to Chariot, which was a single-arm phase 2 study of immunotherapy in a small cohort of 33 patients across Australia. So these patients were stage 3 disease, and they had failed prior AC-based chemotherapy. So they were enrolled into the trial where they received a neoadjuvant ipi-nivo, so anti-CTLA-4 and anti-PD-1 in combination with chemotherapy. And then they received this treatment. They had the remaining tumour removed at surgery, and the level of tumour remaining was scored. And then they went on to continue their anti-PD-1 treatment. So we know the treatment response for all of these patients, and the rate of complete clearance of tumour at surgery was quite low. So the PCR rate was only 24%, which is much lower than what the real world rate is now. But it's important to remember these were sort of really high risk patients who had already failed chemotherapy. And importantly, Shireen Loy, who was the PI on this trial, presented a 3-year survival update last year. And even though it's really low numbers, we can see that patients who had achieved a PCR had a really high survival rate without relapse at 3 years. And interestingly, 100% of the PD-L1 positive patients and 100% of the patients with higher TILs had also not relapsed. So from the trial, we had a choice of at what stage we received patient samples. So we decided to go with the samples that were taken closest to immunotherapy treatment in order to find predictive biomarkers. So the biopsies that we took were from the post-anthracycline treatment, but pre-immunotherapy. And at the time, we had capacity to screen 16 of these patients using the 10x Visium for FFPE platform. So we chose the patients based on where they fell in terms of their treatment response, and these were complete, partial, or non-responders. And so ultimately, we were using Visium to try and look at relationships between TILs and known suppressive cell types. So even though it's Visium and it's spatial and it's single cell, I just will show you first a little bit of the treating the samples as pseudobulks. So this is treating each patient biopsy as an individual sample. And we can see here in this MDS plot that the partial responders do tend to cluster together, but that is not the case for the complete or non-responders in blue and red. Also, we looked at differential gene expression between the 3 responses, and we did see a small number of DE genes. So you're probably familiar with the limitation of Visium, which is that each of the spots that generate the gene expression and spatial information are 55 microns in size. So they usually have around 5 to 10 cells underneath each dot. So you need to perform cell type deconvolution. So we used a published single cell data set from TNBC along with the ENCODE reference data set and performed cell type deconvolution, which is a lot to look at as a stacked plot. There are a lot of different cell types. We haven't gone deeper into subsetting in terms of like Tregs or gamma deltas yet. And unsurprisingly, the only cell type that was significantly different between the treatment responses was CD8 T cells, which were significantly higher in that small number of complete responders. So now what we are doing is trying to find regions of interest across these patient samples in terms of where it's predicted that there were clusters of tumour cells and clusters of T cells. This is just one example of a complete responder. So we can see that it's a little bit hard to see here, but in general, the tumour epithelial cells were kind of separated from the T cells. They didn't really overlap. And then an example of a non-responder here. And you can see down here that there is predicted clusters of epithelial cells where there's not really any T cells. So that was more of like an immune ignored sort of biopsy. So what we are now doing is pulling out those T cell and epithelial dots. They're not single cells, they're just kind of mini bulks. And we're doing differential gene expression between the responders and non-responders on these two cell types. Then we'll be doing neighborhood analysis, looking at PD-1 exhausted TILs and whether there's known suppressive cell types around them. And then also looking at spatially important genes by like measures such as Moran's index. So as I mentioned, it is really a limitation of Visium that it is not technically single cell. And so that's where funding from Cure Cancer comes in. So our Visium data is sort of serving as preliminary data to then design custom panels for techniques with better resolution, such as Merscope or Xenium. So we can then validate on a single cell level the genes of interest that we find from Visium. Then also what we're doing to increase the relevance of the project is we are now starting to collect biopsies from patients who are receiving their immunotherapy at Austin Health. So these are early high-risk TNBC patients who are treated with pembrolizumab. So this is important to get our validate our biomarkers in sort of patients that are treated with the new clinical standard. Then as an extension of this project, we are using another imaging-based technique known as TOF-SIMS. So this is mass spec imaging, and this is a technique that can be used to determine the chemical composition of surfaces, where a beam fragments just the top layer of a surface, and the charged species are analysed through for their time of flight. This determines their mass, and we can then determine the chemical formula of the surface. So some preliminary data from this, we are able to achieve pretty good resolution. So we can achieve 5 micron resolution across the whole patient biopsy, or at higher resolution, 200 nanometers, so subcellular. This is just a representative image showing at one particular peak how we can pick up and image the nuclei of the cells. So all of these sort of sequencing and imaging-based techniques, we hope that we will find potential genes of interest that we can then validate in our preclinical models of TNBC. So these are mouse models that are typically very resistant to checkpoint inhibitors. And if we can find therapies that already exist against targets that we find in our screen, then we can try them in combination with anti-PD-1 in these models, looking at the effect on tumour growth and also metastasis, and the effects on the location and number of TILs. So the ultimate goal is really, can we take all of these really complicated techniques and distill it down into like a simple diagnostic test that can help us make better informed treatment decisions, whether patients will respond to chemoimmunotherapy or not. And if not, can we come up with combination therapies that can convert these non-responders into responders? So I just really want to thank all of the people that have facilitated our access to these patient samples. So BCT, as well as Dr. Belinda Yeo from Austin Health, our lab at ONJ headed by Professor Robin Anderson, the bioinformaticians at Monash who have analysed our data, Yang Liao and Wei Shi, our collaborators at La Trobe, our consumer advocates, and also just all of the patients and their families who have consented to the use of tissues for this project. And happy to take any questions.